Alright, so where were we? Okay. Okay, here we are. Alright, so they're both heterozygous. So we have two different alleles. The Huntington's is dominant, so we're going to give it a capital H. Alright? So for a Punnett square, one of these two, so we'll make this section the males. So for example, this will be the males, sex cells, and this, going down this way, will be the female sex cells, alright? Now, remember, Mendel discovered what's called the law of segregation, which says that the alleles do what when you make sex cells? They separate. So, one sex cell could get the big H, okay? And the other sex cell could get the little h. So the male possibilities are what? Big H with little h. Now the female happens to be the exact same genotype. So her possibilities, when she makes egg cells, it could be also big H or little h. So it's one or the other. So remember, these are sex cells. So the males make sperm cells. The females make egg cells. So in this box here, this would be the possible outcome of this sperm cell over here with this egg cell. So if they meet up, you get two big H's. Now if this sperm cell meets up with this egg cell, you get a big H with a little h. She does. If she's heterozygous, she has a disease. Now, it could very well mean that she doesn't have it at this moment, but Huntington's shows up a little bit later on in life. So if she's been genetically tested and she has one copy of it, she's most likely going to get it. Okay? So here's another possibility. Maybe this sperm cell meets up with this egg cell over here, which gives you that genotype. Maybe the little h meets up with the little h, which gives you that genotype. These are possible zygotes. Now, this does not mean you're going to make 4 babies. This just means what are the odds that the baby will have the genotype little h, little h. So if you look at the Punnett square, there are 4 possibilities. 1 in 4 is what? Is normal. Right? Normal with no disease. The other 3 out of 4 are what? Have the disease. We have the disease here, here, and here. So the question is, what are the odds that a child will be born with Huntington's? The answer then has to be 3 out of what? 3 out of 4. 75%. Pam. No, you're right. I'm sorry. Yeah, the question's not. Sorry, that should say, what are the odds that a child will be born and later on develop Huntington's disease? Okay, you're absolutely right. It shouldn't be worded born, because you're not born with Huntington's, but born with the the genetic determined probability that they will have Huntington's in the future. Yeah. So there's a 75% chance this baby will be born, and that later on in life they will develop Huntington's. That's a good clarification. Pamela DeVera. Pamela A. DeVera. Okay. Now, that's a monohybrid cross. I'm going to show you what's called a dihybrid cross. And you'll see it's just a more complicated version of what I just showed you. So what does di mean? 2. So what if we're looking at 2 characteristics? Now that's a dihybrid cross. Because di means 2. Now, again, the situation is very simple. We're going to assume that each characteristic is controlled by one gene, and it has 2 versions. So how many characteristics do you see up here? There's 2. There's color and length of hair. We're going to make an assumption that the 2 genes, because we have a gene for color and we have a gene for length, we're going to make an assumption that the two genes are not linked. That means that they're on different chromosomes. Because one of the other things we have to assume is a law. And I'll show you the law right here. It's called the law of independent assortment. What that means is that the two genes will separate during sex cell formation. And the way they separate has nothing to do with the other gene, because they're not together. Now, I'll show you what I mean by that as we go through this dihybrid situation, okay? So in this case here, if you think about a dihybrid cross, we have, we start with a female that is purebred black with short hair. We know black is dominant to brown. And in this case, what's dominant? Short hair or long hair? How do you know that? Because it's capitalized. You can see it right here. That short hair is capital S, so that's indicating that it's dominant. Okay? What will happen in this case? Well, this male, is it a male? Yes, a male, can only make what type of sex cell? Well, they only have capital B, and they only have capital S, so the only sex cells that they're going to make contain black and short. If you look at the female has only the allele for brown and the allele for long. So her sex cells can only carry the allele for brown and short. If you put them together, what will your baby look like? Yep, black with
long hair.
Well, what's dominant, short or long? So what will the genotype be? Well, this baby will have at least one big B with at least one big S. Therefore, this baby must be black with long hair. Sorry, short hair. Short hair. I just see you paying attention. Short hair. Okay? What happens then if we take these babies and we get them to mate and have their own babies? Well, let's think about the possibilities. Okay? Let's think about the possible sex cells that they can make. Now remember, in meiosis, when you make sex cells, what do you pass on? How many of the alleles? One of the two. So how many alleles do you see here? Two B's and two S's, because they're two different genes. We can only pass on one of the two. So one B and one S. So, what are the possibilities? Well, one is this big B can be passed on with this big S. There's a 50% chance that I'll get a big B for this gene, and there's a 50% chance I'll get a big S for the other gene. Now, Claudia?
Can you not get this big B and little S?
Yes, you can. So another possibility would be, well, we're gonna go through this all again. Would be big B with this little S. Doesn't it look like a different color?
Can you just do the chart?
We're gonna do the chart. We're gonna do the chart. What's a third possibility? Big B with the big S. Sorry, little b with the big S. Or little b with the... Okay. Now, if you look at the probability then, these are what you're going to get. You're going to get 4 different sex cells. Because there are 4 possible combinations. So, these are the sex cells. If you look at the whole Punnett square, it will look something like this. We're going to go through an example of this, okay? So on the left, we have the male here. And on the top, we have, on the left is the male. Top is the female. These are the genotypes with the phenotypes. Now, well, how many blocks was the monohybrid cross? It was 2 by 2 grid. So this is a 4 by 4 because if you think about it, you have how many possible sex cells? 4 times how many possible egg cells? 4. That gives you 16 possibilities. Let's do one together. Okay? So I have an example here. I know you guys were dying for one. So here's one right here. We're going to look at this example here, okay? So we have a man who has Huntington's disease and is also a carrier for sickle cell. Okay? So remember, Huntington's is going to be the big H, right? Big H. Now, he's going to be heterozygous. And a carrier, remember, sickle cell is what? It's recessive. Sickle cell is recessive. We talked about this yesterday. So in sickle cell anemia, if you're a carrier, that means you have the recessive allele. So that means the N is going to stand for normal hemoglobin, and the little n is going to stand for sickle cell, which is the disease version. But in Huntington's, the opposite. In Huntington's, the allele that causes disease is dominant. Okay? It can't be. A carrier would mean that you have the allele, but you don't show it. If it's dominant, you have it and you show it. Okay? So here's the male. The male is, okay, big H with a little h, and big N with a little n. Now the female. The female has no history of Huntington's. So that means she's got to be normal for that. So she's got two little h's. But it's also a carrier for sickle cell. So she has to be this. Big N, little n.
So she can't get the little n?
No, because it says a carrier,
Okay.
right, for sickle cell. So let's think about the male now, separately. So these are going to be, the top row will make them as the potential sperm cells, okay? Sex cells. Remember, the law of segregation says that the two alleles are going to separate. The law of independent assortment says that the way they separate has nothing to do with the other gene. In other words, this gene over here, H and N, don't affect each other. And if they do, we will deal with that later. That's called linkage. Okay? We'll talk about that one later. But for now, you just assume that they separate independently of each other. So let's think about the possibilities, okay? One possibility is a big H with a big N. So there's one possibility. The other possibility is a big H with a small n. Okay, that's two. Very good, Claudia. You can get little h with big N. And then little h with little n. Those are your four possibilities.
So then with little h and little n be normal? Yeah.
No.
Little n, big N.
Well, hold on. Right now they're nothing. This is just a sex cell. We have to look at the individual. So let's look at the male. So let's look at the female now.
Yeah, but little h is normal, right? Yeah.
Yes. For Huntington's. Yes. So let's look at the female. The female's code is little h, big N, little n. So she can only pass on a little h. But that little h could be with a big N or with a small n. I could fill in row 3 and 4 because here are my, if I think about it, I have two possibilities here. This little h with this big N, and that's over here, or this little h with this little n, and that one's over here. But it could be this little h. It doesn't have to be the first one. It could be this little h over here with this big N. Sorry, little h with that big N. Or this little h with that little n. But that, guess what? It's exactly the same thing. So if you get a scenario where you notice that we have 4 columns, and columns 3 and 4 are the same as 1 and 2, you don't really need to do 3 and 4. Okay? And you'll see why, because it's going to give you the same answer. So let's keep it simple. Because the male can produce 4 types, and the female can produce 2 types, 4 times 2 is 8. We only need 8 boxes. Okay? Doesn't have to be 4 by 4. It would only be 4 by 4 if the female also produces 4 types. Okay? But we can see from her genotype, it's only 2 possibilities. It's going to be a little h with a big N or a little h with a little n. So it's only 2 possibilities. So let's take a look at here's a potential zygote. What would the code be? Big H, little h, big N, big N. This one? Big H, little h, big N, small n. This one? Little h, big N with big N. And this one? Alright. And then this one would be big H, big N, small n, big H, little h. Alright. Little h, big N, little n. Little h, little n, little n. Now, we don't need to do 3 and 4.
Question.
Yep.
Does the capital always have to go first?
I do. Technically doesn't matter, but everyone always puts the capital first. So, now it's deciphering this. So remember, if you have a big H, that means you have what? Huntington's. And if you have 2 little n's, you have sickle cell. Yes, because it's recessive. So, the question is, what are the odds that a child will have at least one disease? So, does this person have a disease over here? They have Huntington's, but not sickle cell. But they have at least one disease. Yes. So this one has Huntington's disease, but not sickle cell. This one has Huntington's, but not sickle cell. This one is normal. Right? No disease for Huntington's, no disease for sickle cell. So that one's okay. What about this one? Normal, no big H, and no 2 little n's. We're okay over here. But you have 2, because sickle cell is recessive. Very good, you'd be a carrier. What about this individual here? They have Huntington's, but not sickle cell, so they have at least one disease. Again, this one has both. So we got 2 diseases. This one is normal for Huntington's, so in other words, they don't have Huntington's, and they don't have sickle cell. It's a carrier for sickle cell. And this one does not have Huntington's, but does have sickle cell, therefore has at least one disease. So how many? 5 out of 8. 5 out of 8 have at least one disease. Okay? We'll continue tomorrow.